Good evening, everybody. Welcome to the Genetic Therapies Research webinar. Just to go over some logistics and housekeeping items before we get started. So closed captioning is available in the menu at the bottom of your screen. This session is being recorded. You will be notified by email when the recording is available. To use the public event chat, please select panelists and attendees from the drop-down menu so others can see your messages. And also feel free to submit questions at any time in the Q&A box. Just a reminder, questions about individual diagnosis or treatment plans cannot be addressed. We encourage you to discuss this information you learned tonight with your care team. So my name is Schyler Kline. I am 34 years old, diagnosed with cystic fibrosis at 6 weeks. I am currently not qualified to take a modulator due to having two rare mutations. So I am the lucky part of that 10% that are not qualified to take a modulator. So genetic therapies are important to me because, um, you know, I've participated in clinical research trials the last 3 years, and it's a culmination of 30 years of research, and I really want to use genetic therapies as a way to provide hope and push science research further. I am excited about this part of research because, again, it's 30 years in the making. I started a clinical research trial 3 years ago and dedicated the time, took the hours and time off from work to uh, to do the clinical research trials. And so I was like, I'm going to become more involved. And I am also very fortunate to be actually in good health. And so as long as I'm in good health, I plan on continuing to participate in clinical research trials. I hope that you walk away from this meeting tonight with hope and more information. And then, of course, you know, with a little bit more knowledge and the courage to um, go to your CF or your clinic, sorry, um, your clinic and hopefully maybe participate in clinical research trials. So we have Kara Foshay and Heather Hathorne. They will be presenting genetic therapies for CF and then participating in genetic therapy trials. Kara Foshay resides in and is a native of Northern Virginia. She received her BS in biology from the College of William and Mary, her PhD in cell biology from Georgetown University, and completed postdoctoral training in the Department of Human Genetics at the University of Chicago. Kara joined the CFF 5 years ago and is a leader on the science team, where she manages the industry-based genetic therapy portfolio. Outside of work, she enjoys food, yoga, and life with her husband, 2 kids, Bella and Ryan, and dog, Ziggy.
Thank you, Schyler. I'm thrilled to be here tonight to give you guys an update on genetic therapies for CF. Give me 1 second while I begin to share my slides. All right. So I like to start every talk I give on genetic therapies with a little bit of biology 101. The flow of genetic information is important for understanding how mutations in the CFTR gene can cause cystic fibrosis, and also important for understanding how genetic therapies can benefit all people with CF. Each cell in our body contains the same genetic code, our DNA or our genes. The sequence for each gene is transcribed into mRNA, a copy of that gene's key information. Next, the mRNA is translated into a functional protein. The CFTR gene encodes the CFTR protein, which functions as a channel to transport chloride out of cells. Mutations that occur in the DNA are also copied into the mRNA and are translated into protein where they may have detrimental effects on the protein function. For example, mutations in the CFTR gene can result in a CFTR channel that doesn't transport chloride. Cystic fibrosis can be caused by many different mutations in the CFTR gene. Hundreds of CF-causing mutations have been identified and occur throughout the entire gene sequence, shown on the left. The location of a mutation in the gene dictates how it will affect the CFTR protein. Some parts of the protein are more essential for function than others, so mutations in those regions will be more severe. Some mutations can cause destabilization of the protein or prevent it from folding correctly. These types of mutations can be treated with modulators, like tezacaftor, which we call a corrector. Other mutations, like G551D, prevent the CFTR chloride channel from opening and closing. In this case, potentiators, like elexacaftor and ivacaftor, can help to keep the channel open and promote chloride transport. But the take-home message here is that modulators don't change or correct the CFTR-causing mutation. Instead, they simply bind the protein and improve its function. But we all know that some mutations prevent any CFTR protein from being made. We sometimes call these stop mutations, nonsense mutations, or PTC mutations. If there is no CFTR protein inside of the cells, then CFTR modulators have no way to act. The good news is that genetic therapies can work even when no CFTR protein is made. Some of the mutations that could benefit from genetic therapies are listed here, but not all of them. In general, these approaches are exciting because they could be applied to any CF-causing mutation. There are three main types of genetic therapies that the Foundation is pursuing: gene editing, gene therapy, and mRNA therapies. Let's talk for a minute about gene editing. Gene editing therapies deliver a protein, such as CRISPR, which you've probably heard about in the news. This allows for a permanent correction of a disease-causing mutation within a person's own DNA. The term gene therapy refers to delivery of a new, non-mutated copy of a gene that makes a functional protein inside of the cells. But gene therapy doesn't change or correct a person's existing mutation, so the therapy can only work as long as the new DNA is present in cells. RNA therapy refers to delivery of a new, non-mutated copy of CFTR mRNA that can encode a fully functional CFTR protein. I group ASOs, or antisense oligos, in here because they're a related therapy using small nucleotide sequences that can bind to and disguise mutations on the mutated CFTR mRNA. These allow for production of a partially and sometimes fully functional protein. You'll find all of these approaches in our pipeline, but gene editing therapies for CF are still several years from entering clinical trials. So for the rest of the talk, we'll focus on mRNA and gene therapy. There are currently 8 genetic therapy trials enrolling people with CF in the United States and abroad, which is really exciting. At the top, we have Splicesense antisense oligo trial, which is only for people with CF that have the 3849 plus 10 KB mutation. This therapy looks good in lab studies, and similar modalities have been used in other diseases, so we hoped that it will be safe and well tolerated. Below that, we see 3 mRNA LNP programs. They're all similar in concept, with differences in the formulation of the lipid nanoparticle and the CFTR cargo. We hope to see data from these programs toward the end of this year, or potentially at NACFC. Moving down, there are 4 gene or DNA-based therapies in trial. All deliver CFTR as the cargo, but they use different viral-based delivery vehicles. AAV, or adeno-associated virus, is the most common vehicle in the broad field of genetic therapy, and this is used by 4D Molecular and Spirovan. Crystal Bio uses an HSV virus. BI at the bottom is unique. They're currently enrolling in Europe, but hope to come to the United States, and they're using a lentiviral approach that integrates a new copy of the CFTR into the DNA of cells. Now that we have several mRNA and gene therapy trials underway, I'd like to highlight some of the differences and similarities you can expect. Even though the trials may begin enrolling those people with CF that are ineligible for or unable to tolerate modulators, both mRNA and gene therapies could be used to restore CF for any person with CF. Both modalities also target the differentiated lung epithelium. These are mature cells that line the airway and are expected to be replaced over time. Because of this, the duration of benefit and the dosing regimen for these therapies are not the same. mRNA is short-lived. It lasts only a few days within cells before it's degraded. Therefore, an mRNA therapy would need to be redosed every 1 to 3 days. In contrast, DNA is capable of lasting longer within cells. It's actually expected to persist for the life of an individual cell, but we don't know exactly how long that is. We expect to see the therapeutic benefit from DNA therapies to last for several months to a year. Current trials are aiming to deliver just 1 dose and will monitor participants initially for improvement and then for any potential loss of function over several months. There are challenges specific to each of these types of therapies, like lipid nanoparticle-associated inflammation for mRNA therapies. And there's questions about the immune system's ability to recognize DNA-based therapies that might prevent effective redosing. But the biggest challenge for both of these is delivering enough of the genetic therapy to the right cells so it can be effective, but also safe. In drug development, we call this safe dosing window the therapeutic window or therapeutic index. So why is safe and efficient delivery such a challenge? One reason is the complexity of these therapies. All genetic therapies, with the exception of ASOs, have 2 parts. The cargo, highlighted in the middle here, the DNA or the RNA, the genetic material that enters the cells and will eventually make a CFTR protein. The delivery vehicle surrounds and protects the genetic therapy as it travels through the body and facilitates uptake into target cells. Now, delivery vehicles come in several flavors. Some utilize the protein coat of viruses with the infectious genome and the ability to replicate removed. Common non-viral vehicles are comprised of lipids, which are fat-like molecules that can create protective layers around the cargo. We often talk about delivery vehicles in simple metaphors, like the truck that transports goods, the bottle that will safeguard your message across the sea, or the wrapper around your favorite candy. But the truth is, the delivery vehicles are an integral part of the genetic therapy, less like this candy kiss and more like a fruit by the foot, where the wrapper is actually wound up within the treat. These 2 pieces, cargo and vehicle, must be manufactured together into a single unit that can traverse the airway and CF mucus while avoiding bacteria, immune cells, and enzymes working to destroy foreign invaders. One question we often hear is, how are RNA therapies different from COVID vaccines? The truth is, they're not very different at all, but the cellular targets of these therapies are different. Vaccines target immune cells, which are found throughout the body, And their number one job is gobbling up things that look foreign, like viruses or LNPs or genetic therapies. For CF, the target is lung cells. And the number one job of lung cells is keeping foreign things out, making CF a much harder target for delivery. Another delivery challenge is the size of genetic therapies. Trikafta, shown on the left, is a small molecule, which can easily diffuse through the body. In contrast, nucleotides or genetic material are large and bulky. Shown here is one RNA nucleotide. For example, each copy of a CFTR mRNA is made up of 6,200 nucleotides, and several of these mRNAs are packaged into each lipid nanoparticle. I like to compare this to the size difference between a kumquat and a watermelon. And I think it's easy to see that small molecules like Trikafta are much, much easier to deliver. Now that we understand how genetic therapies and delivery of these therapies should work, I'd like to explain one difference in the potential risks between integrating and non-integrating somatic gene therapies. In blue, you can see that all of the genetic therapies in clinical trials and under development for CF are somatic cell therapies. The word somatic refers to adult cell types in the body that are not involved in forming a new embryo and therefore are not involved in passing down of genetic material to offspring. In addition, all of the genetic therapies in clinical trial for CF are directly targeting the lungs, shown in green, through inhalation. This means that most of the delivered therapy will stay inside the lung, and circulation or uptake throughout other tissues and cells is less likely to occur. Even though these therapies do not target germline cells that are responsible for passing down of genetic information, regulatory bodies like the FDA and the EMA require drug development companies to look for and characterize the presence of genetic therapy in any other tissue before a clinical trial can begin. For transient non-integrating therapies, which are on the left, like 4D's AAV trial and any of the mRNA trials that are ongoing, any therapy that might be taken up by off-target or non-lung tissues will eventually be lost as the therapy degrades and the cells of the body turn over. This means there should not be any permanent changes to the DNA in germline cells. Only one current clinical trial, Boehringer Ingelheim, is using an integrating lentiviral approach, depicted on the right. This means that the therapy inserts a new and permanent change to the DNA inside any cell that takes it up. BI is using inhalation to target only the lung cells. However, any off-target uptake of this therapy could result in a permanent change to the DNA in that cell. While we don't anticipate an inhaled therapy being delivered all the way to germline cells, the risk does exist, and it will be present in this trial's informed consent. Now, I've shown this drug development funnel before, highlighting the slow and narrowing path from early discovery programs, which we have a lot of in the lab, all the way through approval and market. While the 10 to 12-year timeline may look surprising, I'd remind all of you of the long development path to our first modulator and the acceleration of follow-on therapies once the technology and the regulatory path were established and validated. We hope to eventually see multiple genetic therapies available for CF, but it is still early days for this field as a whole. All of the challenges I just reviewed, as well as the careful examination required for development of therapies, take a long time. So I thought I'd highlight here exactly what biotech and pharmaceutical companies evaluate and what we know about a genetic therapy before it moves into clinical trials. When is a therapy ready for clinical trials? A biotech or pharma company requests FDA approval to begin a clinical trial when the preclinical data gathered in models can provide sufficient evidence that the therapy could be safe and could be effective for treating disease symptoms in people. The goal for CF is to restore close to normal levels of CFTR function in the cells of the lung that normally express CFTR protein. But what does that really mean? Data from our research community have helped us to understand how and where dysfunction occurs in cystic fibrosis and where CFTR restoration needs to happen in order to improve lung function, which we measure through FEV1. We believe that inhaled therapies will need to travel down the trachea and reach the conducting airway, the bronchus and bronchiole, in order to improve FEV1 for people with CF. Within the conducting airway, there are many different types of cells, and not all of them normally express CFTR. Data from our research community suggests that CFTR is normally expressed in secretory cells and ionocytes. We believe that restoration of CFTR, specifically in secretory cells, will be necessary to drive improved chloride secretion and therefore improved lung function. One of the really important tools in our research toolbox are lung cells that are taken from patients. We can grow these cells in a dish to make a layer of cells that looks very much like the inside of a lung. And we use these to test genetic therapies. First, we will often test the cargo. We can measure chloride current in cells to confirm that delivery of DNA or mRNA cargo can make a functional CFTR protein. And in fact, all of the therapies that are currently in clinical trials have demonstrated the function of their CFTR cargo in cells. We can also look at delivery in cells in a dish. We can measure uptake of delivery vehicles by the correct cells using visible proteins like GFP or green fluorescent protein. All of the therapies that are currently in trials have demonstrated uptake by secretory cells. The next challenge is putting it all together, delivering the cargo and vehicle as one integral product, knowing that changes to one may change the other. First, we determine whether the cargo and delivery vehicle work together in cells. Once that's established, cargo and delivery are tested together to see if they're safe and functional in model systems. Now, this looks like a straight line, but to be honest, it's not. This is a cyclic process that requires sometimes years of repetition and optimization before we can say we think it might work in people with CF. So why do we go into clinical trials with some unanswered questions? And the truth is, no single model can replicate all of the barriers that a therapy will encounter when delivered to a person with CF. And no two people with CF are the same. Some people will always respond differently than others, just like some people respond better to modulators than others. In addition, in people, we can't always measure the same things we measure in cells or models. FEV1 is a good example. This is one metric that we cannot measure until a therapy goes into clinical trials. One important note is about bronchoscopy, and Heather will highlight that some of these trials do involve bronchoscopy. We know this is an invasive procedure for people that are enrolled in a trial, but it's also an important measurement. One thing that we can measure in cells, in models, and in people is expression of CFTR protein. Even if a therapy is not successful in the clinic, Trial data, like bronchoscopy, can help us learn and improve our models so that subsequent studies will have a better chance of success. With that, I'd like to say thank you for your time. I hope you've learned something new, and I'm excited to answer your questions in a few minutes.
Thank you, Kara. That was amazing. So now you had the science, you saw the science behind genetic therapies. And now we're gonna hear from Heather in regards to actual genetic therapy and what goes behind the trials. So Heather Hathorne resides in Homewood, Alabama, and is a research coordinator at the University of Alabama in Birmingham. Heather is a respiratory therapist by training and has been part of the research team at UAB for over 20 years. Heather enjoys hiking, camping, and exploring the U.S. national parks.
Thank you, Schyler, for that introduction, and thank you, Dr. Foshay, for that really nice explanation of genetic therapies. And I'm excited to be with you today to just kind of walk you through what participating in a genetic therapy trial may look like and help you learn more if you want to get connected. Next slide, please. So if you're thinking about a genetic therapy study, you may have a lot of questions. Questions like, what's the dosing going to look like? How many times am I going to have to come to the clinic? What's going to be done when I do come to the clinic? And these are all great questions, questions you should be asking. As far as dosing and visits, that's really going to depend on the specific drug, and every protocol is different. There's a lot of routine assessments that are typically part of all clinical trials, and genetic therapies are no different. Things like vital signs, height and weight, blood draw, EKGs, pulmonary function testing or FEV1, maybe sputum induction. And then there's other assessments that may be a little bit more unique to genetic therapy trials, things like bronchoscopy. And Dr. Foshay explained why those are so important in genetic therapies, and other measures like chest CT scans. Next slide. So travel and reimbursement, that's another really important question. If you have to travel to the research site for this type of visits, travel is usually arranged by the research site and the sponsor, and reimbursement is usually provided for travel and meals after the completed study visit. And compensation, which is for your time, is usually provided for each completed study visit after that visit. And this process of what this really looks like and how long it takes is really going to be different from institution to institution. And then we know there are a lot of challenges when participating in any study, like a genetic therapy trial. The time commitment is long. We acknowledge that. That can be challenging. Travel itself can be a challenge. And sometimes sponsors change the structure of a trial. And what I mean by that is you may be enrolled and participating, and then these things come up that we call amendments, because as we go along and we learn more, perhaps something is changed or a visit is added or a assessment is added, and that changes the schedule, and maybe you have to pivot, and that can be challenging. Next slide. But all of these questions are always going to be explained in the informed consent. So let's talk a little bit about that. What is that exactly? Well, the informed consent is the document that's going to provide all the information about the trial. And the study team will review the consent with you before you participate. And we really encourage you to involve your family or your care partner in the consent discussion. And if you don't understand something, you should certainly ask. We want you to ask questions. And remember, you can take the informed consent home. You should do this. You should think about it. Review it with your friends and your family, your CF care team. It is okay for this to take time. You shouldn't feel rushed or that you have to make a sudden decision. And only when you feel like your questions have been answered, that's when you should sign and date the informed consent. Next slide. So what's your role as a participant when participating in these types of therapies? Well, follow the study plan. That's really important. Consistency is important. Keep your study appointments. You may be asked to complete diaries, questionnaires. You want to always bring those back, maintain any kind of dosing schedule. Communicate with us. It's important to keep us informed. Let your study doctor know how you're feeling, if you have concerns, if you have symptoms or side effects. And before you enroll, you should know who to contact. When should you call the research team? And when should you call your clinical team? And that'll be explained to you during the informed consent process. And remember that an informed consent is not a one and done. It's an ongoing thing. You have the right to withdraw your consent at any point in time, and in no way would this impact your CF care that you receive. And remember that your safety is always the priority in any CF trial. Next slide. So speaking of safety, how will your safety be protected? Well, the FDA has strict guidelines and safeguards in place, and the CF Foundation adds additional safety measures for studies in CF. Safety is always the priority. And what are the risks? Well, risks are going to be different for every clinical trial because they are different. You may experience side effects to medications or treatments. You may receive a treatment that doesn't work for you, but the informed consent will explain all of these study risks so that you have time to understand and ask questions. And this diagram explains a little bit about safety in clinical trials. And if you would like to learn more about this, I encourage you to visit cff.org/safety. You can click into each one of these layers and each one of these groups and understand what their responsibility is and what their role is in monitoring safety. Monitoring safety goes on before enrollment and during the clinical trial. So check this out if you want to learn more about how safety is monitored in a research study. Next slide. So let's talk a little bit about some of the current enrolling genetic therapies. And I just wanted to give you a very high-level overview of some general information and trial requirements. So let's start with mRNA therapy. And that's when that new correct copy of mRNA is delivered into the lungs. And these studies are usually for patients not eligible for modulators and sometimes include patients who are unable to tolerate modulators. The FEV1 requirement is going to be different. It ranges. Usually, it's not uncommon to see it between 40 and 90%. These therapies are multiple visits that are completed over several weeks. They are inhaled. They're frequent dosing, daily to weekly. And the follow-up is usually around 6 months. And the current mRNA therapies that are enrolling include the Vertex 522 study, Arcturus, and the RECODE trial. Next. Let's talk about those gene therapies or non-integrating. This means they are not permanent. This is when that new correct copy of DNA is delivered into the lungs. Same thing, these studies are for patients who are not eligible for modulators. Some include patients who are unable to tolerate similar FEV1 requirement. These are multiple visits completed over several months. So these studies are a little longer. They are inhaled, one single dose. Now, let me point out that does not mean one therapy and it's done. These non-integrating drugs will still need to be redosed. They are not a permanent therapy, but these current studies are looking at a single dose first in order to determine and figure out what the right redosing schedule will be. The follow-up requirement is a little bit longer, up to 5 years, and the non-integrating gene therapies that are currently enrolling include the 4DMT study, Spirovant, and Crystal Bio. Next. Let's talk about those ASO therapies. Remember, these bind to that faulty CFTR mRNA and help the cells make that full-length CFTR protein. Now, these are different. They're designed for very specific mutations because they carry very specific instructions. Same FEV1 requirement, around generally 40 to 90%. These are multiple visits completed over several weeks, inhaled multiple doses. The follow-up is a little bit shorter, weeks to months. And there is one ASO therapy currently enrolling, and this is the SpliceSense study. And this is specifically for the 3849 splicing mutation. Next slide. So let's talk about how you can get involved. If you would like to participate in a clinical trial, there's many ways you can get involved. Start with asking your CF center. Talk to your provider about participating. Ask to speak to your research team. Perhaps you could use the clinical trial finder to search for clinical trials that interest you, that may be being conducted at a different CF center. You can talk to your provider about participating in these studies as well. And if you're not sure about where to begin or you need help, you can contact our clinical trial navigator. You can reach her. Her name is Katie Howell at trialnavigator@cff.org to learn more about clinical trials and the resources available. And don't forget that you can also join Community Voice to help shape the future research projects. We want you to make your voice heard and ensure that CF research meets the community's needs for all people with CF. Next slide. So if you have questions about clinical trials, let's talk about the clinical trial navigator and how she can help you. She is a former CF research coordinator, so she's familiar with CF research, and she can provide personalized one-on-one assistance to people with CF and their families who have questions about studies. She can help you understand the clinical trial process. She can assist you in using the clinical trial finder, and she can facilitate contact between you and the research site. So you can email Katie at trialnavigator@cff.org, or you can visit cff.org/trialnavigator to get started, or you can scan the QR code. And you'll hear back from Katie within 2 business days. Next slide. Let's talk about the Clinical Trial Finder. What is it? Well, it's an incredible resource that's provided by the CF Foundation that allows you to search for clinical trials you may be interested in. And you can find the Clinical Trial Finder at cff.org/finder. There's multiple ways to use the Clinical Trial Finder. If you're not familiar with it, there is a tutorial video found on the Clinical Trial website, or you can contact Katie, the Clinical Trial Navigator, and she will walk you through the entire process. Next slide. If you do check out the Clinical Trial Finder, this is a snapshot of what it looks like. You'll notice there's a filter box. You can search here by age, location, mutation, and more. You can find studies that are currently enrolling. You can even view study results. You can contact a research coordinator directly through the Trial Finder and express interest in a specific trial. Or this is really cool, you can sign up for email alerts to be notified when new studies are posted. So if you haven't checked out the Clinical Trial Finder, I really encourage you to at cff.org/finder. Next slide. So what if you want to participate in a trial at a location other than your care center? That's not uncommon because genetic therapy trials sometimes only occur at a really small number of care centers across the country. When that happens, we call that a clinical trial referrals. That's when you are participating in a study at a different location than where you receive your care. And you may ask, how is that possible? Can I do that? Or would I have to change the location of where I received my care? Absolutely not. You will continue to receive your regular clinical care from your usual care team. And you may wonder, well, how will the research team and the clinical care team stay connected? We'll stay in close contact through your study participation, and we'll discuss this with you during the informed consent. You can invite your care team into that discussion. We can discuss this via Zoom if necessary, and we can come up with a plan that you're comfortable with for how we all can stay connected. Next slide. So let's go on a road trip and discuss what a study referral may look like. We call this our referral roadmap. So the first stop on our journey would be to identify a clinical trial. You can do this by using the Clinical Trial Finder, or maybe you were a referral from a care center. Next. So your next stop would be to get connected. Your care team can help you do this, or you can contact the research team through the Clinical Trial Finder, or you can reach out to Katie Howell, and the Clinical Trial Navigator can help you get connected. Next. The next stop on this journey would be to determine if you're interested in the clinical trial and if you're eligible. So this is when the research team will contact your care team only with your permission first. You would sign a medical release to have your medical information be sent to the clinical research team. This is where we'll go over all the inclusion and exclusion of the clinical trial and talk to you about your CF history, the medications you take, and determine if you are eligible. Next. And the final stop on this road trip would be to enroll. After we've determined perhaps some basic eligibility and interest, that's when we would really start diving into these informed consent discussions. And if you are remote, as in the research site is a ways away, we can talk about the informed consent via Zoom. You can invite your care team. We can talk through the risk, the benefits. We can answer all of your questions. We can give you time to think about it. And remember that your participation is voluntary. And you most likely, if you are a referral, would have to travel to the study site for the research visits. That will be set up for you, and the care team and the research team will stay in close contact throughout this process. Next slide. And I do want to point out that this is not a one-way highway with no exits. You can exit this process at any time. So don't feel like that if you initiate contact, it's going to commit you to go through all of these steps. At any time you feel like, thank you for this information, but this study is not right for me, that is your decision to make. And so you can exit the road trip and you can, decide not to move forward. That is okay. Next slide. So we've talked a lot about genetic therapies, but in closing, I do want to highlight the REACH study. If you're interested, you can be a part of the future of cystic fibrosis treatments. If you have CF and you are not taking CFTR modulators like Trikafta, you may want to consider joining the REACH study. There's no drugs involved in this, but by sharing your health information, including things like blood samples, you can contribute to research to help develop new therapies for people with CF who don't benefit from modulators, new therapies like genetic therapies. Participants will receive compensation, travel costs are covered. People who enroll in REACH will be enrolled in the study for up to a year, but you can stop participation at any time. You will have a study visit when the study starts, and then again at 3 and 6 and 12 months later. And you can learn more about REACH by scanning the QR code or visiting reachcfstudy.com. I do want to point out that studies like REACH are crucial to help us and inform new therapies like genetic therapies so that we can advance these trials for all people with CF. And with that, I think I will invite Schyler and Kara back for questions.
Thanks, Heather. It was awesome. So yeah, I wanted to emphasize definitely the importance of, never hesitate to ask questions and know that even though you commit to a clinical trial, that you can leave the trial at any time if for any reason. I've been participating. I was in the VX 522, which is still continuously going. And just understand that it is a process, and patience is definitely required. It's always like a thing of, yeah, like, well, does it work? Like, will it work? Well, it's a trial. So, again, that patience, um, constant communication and contact with your research team. And I also want to emphasize that I've always been lucky where my research team is always in contact with my clinical team. I actually went to National Jewish Health is where I went to, and they were outstanding with that. I am no longer in Colorado. I am now an Ohio resident. So, what Heather was talking about, travel and stuff, is these clinical trials. They want you to participate, but just know that there's definitely options, and to definitely talk those over with your clinical team and of course the research team. There's also always challenges too. But again, your research team, clinical research team, or clinical team is always there to support you. All right. So we're gonna open up to Q&A, and just give me a minute here. So the first question is, what are some of the key concepts people with CF need to understand prior to participating in genetic therapy trial? Um, you can always ask for your research team for the consent form. Like Heather was saying, take your time, talk to it about with your family and friends. Don't feel that you have to rush and get it signed and immediately in. Pick it apart, look at it, and if you have questions about any part of it, again, don't hesitate to ask your teams. Next question is, are these genetic therapies a one-time fix? Will they benefit any other organs outside of the lungs?
I'm happy to take that one. Genetic therapies that we talked about here today are unfortunately not a one-time fix. mRNA therapies will likely be dosed between every 1 and 3 days, and that would repeat for the lifetime of a patient, just like modulators are taken daily for the life of a patient. Right now, the genetic therapies that are being developed and those that are in clinical trials target only the lung. And the reason that they target only the lung is because, one, this is the main source of infection and complications with CF. So we see it as the most important first target, but also because delivery of these therapies is such a challenge. As I highlighted, these are big and fragile and difficult to deliver to the correct cells. So we started by really focusing in on what we thought would be the most impactful target. We hope that one day, once genetic therapies for the lung have been established, that we can move on to identify genetic therapies that could benefit other organs. But for now, they'll be lung only.
Perfect. Next question. Can you explain the difference between genetic therapies and CFTR modulators? Will people who take modulators also be able to benefit from genetic therapies?
Yeah, absolutely. So modulators are small molecules that are taken systemically, diffused throughout the entire body, and once they're inside cells, they bind to CFTR protein. Genetic therapies deliver the genetic material necessary to make a new CFTR protein. So genetic therapies could, in theory, benefit anybody with CF, regardless of what mutation they have. Trials are going to start for people that are modulator ineligible because that's where the medical need is the greatest. If and when these therapies become approved, they may be available for people who take modulators.
Perfect.
Yeah.
Next question. Regarding those drugs in phase 2 in the pipeline, how close are they to completing phase 2? And are you optimistic?
I guess that one's for me too. So these programs continue to enroll and they aren't finished. And the population they're enrolling is small. And also these trials are proceeding very slowly with constant review by oversight boards like the Data Safety Monitoring Board. So it's impossible to predict exactly when these trials will wrap up. And it's impossible to make decisions without a complete data set. These are small trials, and we need data from all the patients at all the time points before we can really assess whether or not they're working or not. We do hope for readouts in the next several months to year, but unfortunately, we don't have a lot of control over that. We are optimistic, though, for learning. I think we're going to learn a lot in the next year.
And I'm going to add to that, Kara, is as a person with CF and participating in these clinical trials, I am remaining optimistic. These clinical trials have almost kind of personally provided me with this kind of this sense of hope. So
I love that. Thanks for sharing.
yeah. Next question. With such a small population of those with CF that can be in these studies, because only those not on modulators, won't it take a very long time to complete these studies and get enough data?
Yes, it will. And that's why they've been ongoing for as long as they have. It's a small population. And these trials are actually quite costly to run compared to a modulator trial. And so they're not necessarily being offered at as many sites. So they will be slow, they will take a long time to enroll. And like I said, we really need that whole data set to be able to analyze the true effect.
Perfect. So this next question is actually addressed to me. Why did you select VX522 clinical trial over AAV gene therapy? So I think it really depends on your clinic. So National Jewish was one of the ones that were selected for the VX522 clinical trial. And so, my research team came to me when I was going into clinical trial, adjusting clinicals. Um, Visiting my clinic for my yearly, and they're like, "Hey, we have this upcoming, trial that you are qualified for because you're not qualified for a modulator, and you have two rare mutations." And they kind of basically explained the science to me, and I was like, "Well, that's 30 years of research, and I would, yeah, like." Give me the consent form. I'll review it, and then sign me up. Actually, to be honest, this is the first time I've actually heard of the other ones. So VX's course is one of the biggest ones with Vertex Pharmaceuticals. So next question: do they need non-CF patients in trials for safety?
I can take that. Some of the trials did begin in healthy volunteers. Some will not, and that's due to the nature of the risk associated with genetic therapies as really a new modality of therapy. So while some of them may start in healthy volunteers, the RNA trials definitely started in healthy volunteers. The Splicence trial did. The 4D molecular one did not. And many of the viral therapies will not start in healthy volunteers.
Perfect. Next question: what mechanism is helping the viral vector make it through the mucus to deliver the gene therapy to the cells?
That's a great question. It turns out that viruses have evolved for millions of years to be really, really good at getting through mucus and into lungs. And so really the goal of using viral capsids to deliver gene therapy is to take advantage of evolution and get something that's already really good at its job. In fact, there are studies looking at using viral proteins on non-viral delivery vehicles to see if they can boost up the efficiency and help them get through the difficult lung environment.
Perfect. All right. So next question: how can a person with CF know the best time to enroll in a genetic therapy study? If I enroll early, will I miss later options?
Yeah, hi, I can take that one or jump in first. Because all of these therapies are different, if you enroll in one therapy, it may delay when you could enroll in another. And so I think that's why it is so important that you understand your particular mutation and how it works or how it doesn't work, and have those conversations with your CF care team and your CF provider so that they can help you understand what is the best therapy for you and when you should enroll in what trial.
Perfect. And again, that's definitely a question for your research team, or even your clinical care team will also could answer that question as well. Whenever I went to go visit my clinical care team, they would, of course, my doctors would come in, but then they're like, okay, well, see, research kind of is heading down. I'm like, cool. So, all right, next question. Are there any trials internationally or for children, even the REACH CF study?
I can try and take that one. There is clinical research going on abroad, and this is where I would really encourage you to check out the clinical trial finder, because you can find all the information about what studies are available and what you may qualify for there.
Perfect. All right. Next question. If you're on a trial, are you excluded from others and for how long? Like, is it one choice?
So usually when you're participating in one study, you can't participate in other trials. And sometimes some therapies or some clinical trials are going to have a washout period. And so that means you would have to wait a certain amount of time before you could enroll in another trial. But again, they're all going to be different on that washout period and unique to the very specific mutation. So all that information is going to be in the protocol or in the informed consent.
Perfect. All right. And this will be our last question for the evening. What are you most excited about in this area of research? Everyone can answer this. So, did we want to start with Heather?
Sure.
Okay.
I'm super excited. The science is moving really fast, and it's just such an incredible and exciting time to be a part of this community. And I'm just really excited to see what's in store and what we'll find out. And so it's just really an incredible time. Just happy to be a part of it.
That's a great answer, Heather. I'm also really excited for the learning, and Heather is right on that this field is moving fast. I think all of these are really interesting, but I will say that I manage our gene editing portfolio, and so I am really excited. I'm just back from a conference on gene editing and really excited to see the advances in that field. A few years ago, this field didn't exist. So the fact that we have the first FDA approved editing therapy for sickle cell disease is super exciting. And I'm excited to watch how fast that progresses for CF.
Perfect. And as a person living with CF and not qualified for modulator, again, this kind of research and providing this research up front is, again, provides a sense of hope for those that are not qualified to take a modulator for various reasons. And again, this is I was diagnosed in the early 90s, and you would there 18 was the time, like that was. And now 30 years later, the science makes sense, and it's exciting. So thank you, Heather and Kara, for sharing today.
Thank you, Schyler.
Thank you, Schyler.
All right. That is all we have for this evening. Again, if you have any additional questions, please feel free to reach out to the CF Foundation and get more involved.